you. So this is the third of these sessions we've had, and we've had wonderful audiences at all of these, and I appreciate that you've come and all of you people doing the work behind the scenes to make it happen. Thank you. So I do come from Western Australia, but I was born in Cessnock, so it's sort of like I've been on both sides of the continent. So following Colin, I am talking about plants too, and plants interacting with the soil and with the organisms in the soil. That's what it really is all about. So I'm going to be talking about plant-soil interactions and soil health. You've had a big discussion about what soil health is. I'm going to talk about soil biological fertility, plant diversity, inputs, and then touching on measuring all of this and the Soil Health app. Now this little logo here is the Soil Health app, which we developed in the last year with funding from the National Land Care Program. And it's free. The app is free. Download on either of your phones. Don't download it now because there's a lot of data, or not data, but information that you download. So when you've got your Wi-Fi on later, and we can talk about it afterwards if you like, about what more is on that. But okay, we've moved forward. Now, which button am I pressing here?
Yeah, this one.
That one. Okay, thank you. So that's getting going. These little springtails came from a farm near Pingelly in Western Australia, and oh, they're not doing anything. I'll just make them work again. So just use the videos, because in your soils there's a lot of action, and the animals are helping to create a lot of that action. So I like to use these. So in my career at the University of Western Australia, I've been funded by a lot of different organisations, and maybe you've partly funded me. Did you think that was a possibility? So I started at UWA funded by the Australian Wool Corporation, as it was called then. Now it's got a different name. And we were working on soil biology in pastures. So the whole point was to investigate soil health in those days. We didn't use the word soil health then. But I've been funded with a team of people at UWA and elsewhere to look at what is going on in soil. And actually, 49 years ago, nobody really cared much about this. Soil biology wasn't on the radar. We didn't have soil biology forums. It just wasn't there. So struggling through my career, I've been working against the grain a little bit, literally, and thinking about what's going on. I'll come back to that in a minute. So what I want to start with is soil diversity, because soils are all different. And we've got sands in Western Australia. Sands aren't just sands. I've learnt that from the pathologists over there. Plant diversity, and then within that we've got microbial and soil animal diversity. I think this is going in and out. I'll just yell. I'm used to yelling anyway. We've also got different soils here. So I've got a picture of sand. Now in Western Australia there's a lot of horticulture on the very sandy soils around Perth. I've got a picture of clay, and that came from northern New South Wales because it's a really decent soil compared to what we have. But the soil type, I'll maybe touch on this later, but if you're trying to compare your soil with or your farm with somebody else's and you're doing measurements on soil biology, actually these things make a big difference to what you're going to get. So when I talk about your soil biology, or when you think about it, you think about your place. Don't worry what's going on in somebody else's, because it's about your own soil. Which reminds me, you have all got soil. Yeah? Have you got a spade? Do you use it? Yeah. Well, actually, spade is probably the best tool that you've got for thinking and looking at soil biology. And you are on your land. You can dig in the soil at different times of the year, different paddocks, and you actually have a huge amount of information that we can't override. So remember that you're in charge of this. So soils have got mainly mineral stuff and only a little bit of organic matter. But that organic matter drives the healthiness of the soil. Now, Colin's already talked about plant diversity. Most different plants have their own community of microorganisms. There's a lot of overlap between them, but the plant diversity can have an impact on the microbial diversity. Now, while there's a lot of diversity of microbes and soil animals in the soil, a lot of them do the same thing. So it doesn't really matter if you've got different species, because a lot of those species overlap in their function. So in the soil, we have hyphae from different fungi, hyphae in the roots that spread out into the soil, fungi, bacteria and fungi that may or may not be able to grow in culture in the lab, lots of different animals, rhizobia in your legumes, little animals again. But in the scientific community, we have experts on all these things. So we have an expert who knows all about the rhizobia. Someone else knows about springtails. Someone else knows about mycorrhizas. Someone else knows about bacteria and nutrient cycling. That's a science. All these little experts in a science community, but you manage the whole thing. You don't need to know all the details of all the little bits, but in fact, on a daily basis, you are managing this complex system that's going on in your soil. Do you feel confident about that? Yeah? Can't hear an answer. But what I'm saying is you don't have to know all the little things that are all the intricacy of it, but you need to feel confident about you're in charge of managing this whole universe, really. So we've heard this already in the talk, in the discussion that Oliver led. So chemical fertility, biological fertility, and physical fertility. When I first started doing research, and I look up in the literature about fertility, these terms weren't used then. It was just fertility. You had a fertile soil, basically because it had enough nutrients to grow a plant. And that was really, all the publications talked about soil fertility. And that was a problem for me, because I'm in a plant nutrition, soil science department at UWA, with a lot of experts all around me, and I'm a soil biologist. How do I fit into soil fertility? Because it's really about nutrients. Okay, microbes are related to nutrients, but how does this stuff about soil biology actually fit into soil fertility? And that's what I was teaching. And I spent a lot of time just thinking about this, because at the end of the day, the agronomists were driving either with selecting the right plant for the right place, getting all of the planting time sorted out, putting the nutrients on. That was so well understood. So how do you squeeze in biology? Now, certainly with the rhizobium, that was part of the package. But the rest of the biology, the fuzzy stuff about all these organisms doing their thing, how do you put that into the decision-making of soil fertility? And biological fertility was not mentioned in the literature until we started to use that word. That sounds ridiculous now, but let's say we introduce the concept of soil biological fertility, and then unpack that. So you've got physical, chemical, biological. But actually, when you then start to define these, you realise that a lot of the biological processes relate to chemistry. A lot of the biological processes relate to physics. So soil aggregation, structure of the soil, aggregation we talked about, sticking things together, the hyphae, the gums. So how do you actually separate these? And the reality is, we already had it spoken to us this morning, they're integrated, they're all connected. So here I was saying, how does biology fit in? Well, it does fit in. It fits into both the physics and the chemistry. But if you don't mention it, you can stick with the chemistry. And I think that's the issue about understanding how they related, and you need to take account of all the three. And I do have another thing to point out. is that some of this biology can be overridden by a focus on the chemistry or the physics. That's the point, really. Because if you get the nutrients right for the plant, you'd say, oh, that's fine. But what about the biological contribution that's made in the fertility? You mightn't get that right. So you need to have all three considered, but they're related. They can't all be defined independently of one another, which is good. So it's a story rather than just a definition of them. So say we get a focus more on biological fertility, then we have to think of time, because it's not a magic thing. You can't just do something and you'll fix this biological fertility, because living organisms, they take time to do their thing, and you can stuff it up pretty easily, by the way. So you need to have time considered, and this will develop with time. And when I first used to do the workshops for farmers in Western Australia, they were telling me this thing about something happened in 3 to 5 years after they changed their practice. And I thought, well, what's that? But they knew something was happening. They didn't know what. So time, and remember we can undo the benefits, so that's also a time factor. We can have nutrient replacement from these processes. We can change the supply of nutrients. So we're not front-end loading the nutrients at the beginning, or they may be added during the season, but that big boost at the beginning, it won't happen fully from biological fertility. So there's a time difference in the scheduling of nutrients. And this has implications. So it has implications for plant physiology. So a plant that's front-end loaded with nutrients is going to grow in a different way and function in a different way to one that has a more gradual supply during the season. It could affect the quality of the grain and the forage. So the physiologists in agriculture basically didn't focus on slow time delivery, except there's a lot of research on slow delivery of fertilisers, but mainly they haven't focused on that. And then costs, and I'm not a farmer, so I can't pretend to deal with this end of it, though we do have some research with economists at the moment. But Colin's already dealt with this. So it can be profitable, and I'll take the word of someone who has to actually spend the money, not me. So I'm saying it can be profitable to focus on biological fertility and try and get that balance. So here we go. Soil is interesting. When I used to give my lectures on soil biology, I always asked students to think about what it was like in soil. So I'm asking you now. What do you see when you're in here? It's dark, you can't see anything. But this is a sandy soil. Hyphae are helping to bind these particles together. So if it's a more clay soil, there'll be less space, the smaller pores, so very different. But the organisms that are gluing all that together, in combination with organic matter, because the organic matter is crucial, is what creates that environment that is all sticky and healthy. So let's look in that space. And in a pasture, you'll have mixed species. So we've got it here, grasses and legume. And the roots of these plants are all intermingled, so they're bumping into one another. The communities of organisms might be slightly different on the different species, but in the soil, it's a mass of roots, different plant species, and then the fungal connections between these plants occur, the bacteria doing their thing with organic matter, and within that community, you've got these plants leaking carbon into the soil. And that is the rhizosphere. So the rhizosphere is that area that's influenced by the roots. And plants, they're like a drain. They drain carbon into the soil. And it can be a lot. It can be 10, 20, 30, 40% of the carbon fixed, feeding the organisms. Now, in the soil, there's different groups. So around the roots, you've got a rhizosphere community, and that's very broad, lots of different types of species. But within that rhizosphere, you've also got pathogens, and pathogens are normal components of the rhizosphere. So these organisms that have potential to cause disease are just part of the community, and the aim is to keep them in their spot. And so some of the rhizosphere organisms can suppress disease, and there's a lot of work that's done on this. So when it's in balance, these don't get carried away and cause a problem. Then you've got mycorrhizal fungi. So mycorrhizal fungi occur in the roots of nearly all agricultural plants, and most of them out in the forest area as well. But different species occur, and they all happily go into most plants, except for a few. What are the plants that won't form mycorrhizas? Yeah. And so they have other strategies for getting nutrients. So the mycorrhizal community is very interesting because roots let these fungi in. They don't say you can't come in. Whereas for pathogens, most plants keep pathogens out. So pathogens can be specific for a particular plant, and the mechanism of keeping the fungus out or the bacteria out is switched on and doesn't let them in unless it's the right fungus or pathogen for the plant. But mycorrhizal fungi have developed a relationship between the plant and themselves, and the plants let them in, which is quite an unusual case. Now with rhizobium, if you grow legumes and you pick the rhizobia that you need for your particular legume, it's specific, which is very different to the mycorrhizas. You get the right legume and you put it on the right, you get the right rhizobium and put it on the right legume. So there's a lot of things going on in the rhizosphere. There's a bit less going on beyond the rhizosphere, but if you think of what Colin said about growing plants in the soil, in fact, most soils in really healthy situations are rhizosphere soil. So the more that plants are involved in colonising, growing in the soil, you've actually created that rhizosphere for most of the soil. Now, it differs with depth as the root becomes less dense, but you really have to have most of your soil as a rhizosphere soil. Now, Gupta from CSIRO, do you know Gupta? Some of you do. So Gupta made this poster. We did some work with him on soil fauna many years ago, and this is a really nice poster which you can't see from a distance, but we can make it available to you. So basically, it's a story about what happens when organic matter goes into soil, and remember most of the organic, or a lot of the organic matter is roots. It's already in the soil. So when it goes on the soil, it kick-starts the chain reaction. So you've got the organic material either as the plant material or as exudates, kick-starting the bacteria and fungi, then all the other animals feed off this process down to the larger organisms. But it's a dynamic thing. It's not all going on at once. Different organisms are active at different times. And the little red buckets relate to the nutrients that are released from those processes of the interactions with the organisms. And how they interact is either they eat each other or they poo in the soil, or whatever happens, the nutrients get into these little buckets, red buckets, and this is the cycling. So it's not all about the bacteria, it's about the whole system that's been created by the supply of nutrients and carbon from the plants. That's what drives the whole system. Now, when you go looking for organisms, we did a study with Gupta out in Western Australia, looking over time, counting little animals. And if you go at the wrong time, you can't find any. So in the summer, you can't find them, but their life cycle is set up so they appear when it starts to rain. And in this study, we were looking at adding different residues of plant, so wheat and lupin. But the point is really here that if you're looking for organisms, they might not be there or you can't find them. Doesn't matter. It doesn't mean that they don't exist. So some are active at some times, others are active at other times. So when you measure something, it mightn't be there, but it doesn't mean it's not hiding away. Now we've looked at grazing, and this was an experiment looking at grazing in terms of mycorrhizas, how grazing affected the colonisation of roots by these fungi. Now we use scissors. Scissors is not exactly the same as animals eating, but we were following the roots of plants when they were grazed or cut. And when plants are growing like this, ungrazed, then the roots have got a lot of sugars in them. When you cut the plants, you also cut the roots. So if you're grazing the tops, but you're also doing something to the roots. Now, not all plant species respond to this in the same way to grazing. Some of them are affected in different ways. Some shed their roots when they're grazed heavily. So, but in this case, there's not a lot, even the roots of the plants which have been grazed heavily have not got much sugar in them, even though they're there. So the concentration of sugar in that. So there's a big impact of grazing, on supply of carbon to the soil, on how that affects roots, on how that affects communities of organisms in it. And we were studying mycorrhizas in this particular study. And this is an example of some work we did with another farmer, introducing perennial grasses into our pastures. Because in Western Australia, in most of the wheat belt where they do have sheep, annual pastures are the norm. So we're trying to put some perennial grass species into the wheat belt, and at the University of Western Australia farm we've been doing that work, which it works, it can be done. Now we've done work with, are there any dairy people here? Nope. So we've done work in dairies south of Western Australia, where in this case with South West Catchments Council we had hectare plots that they'd set up with different soil amendments. I'll talk about that in a minute. Then we were studying the biology of these soils with the different amendments. Now, jumped into DNA. How much time have I got? Okay. So DNA. Have you got any DNA in you? Yeah. So all the bugs in these communities, they've got DNA too. So that the medical people got onto all this technology, and so the agricultural people followed it. The soil scientists eventually followed it. So we can measure, take samples of the soil, get the DNA of the profile of organisms in the soil, and start to think about who's in the soil. And the reason this is quite helpful is that most of the organisms that live in soil can't be grown, like the bacteria and fungi, can't be grown on agar. So in the past, you know, we'd be working with organisms that we couldn't grow. But now we don't need to grow them, we can identify them with DNA profiles. But in this case, the DNA sequencing is collected from a whole community of the organisms in the soil. In this case, it's a bacteria. And then the sequences in that DNA can be related to an organism, or they can be related to a protein that's produced. And that protein might be an enzyme, and that enzyme might be involved in carbon breakdown. So the bonds in the carbon molecules chop, 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 need different structures of the carbon get broken down by different enzymes. And in this study, this is looking at the enzyme profile of the DNA of all the organisms, all the bacteria that are in the soil. And down along here, these are different enzymes that have been detected. And then here, these are different carbon compounds, molecules. So starch, hemicellulose, cellulose, chitin, lignin. So these decrease in their complexity, these molecules. And then these are the organisms that can bust them up. So the colour coding, is anyone colour blind here? No, except we've got one. So in this experiment, the farmers added manure, compost at 2 rates, and fertiliser only. Now, Notice that there's fertiliser applied to all of these treatments, and none of the fertilisers was reduced. None of the fertiliser applications were reduced. Now, we wouldn't normally do that. We would like to reduce the fertiliser to see if the amendments could take account for some of the nutrient input. But the farmers in this case weren't game to let go of their fertiliser input. So the fertilisers went into across all of the treatments in this case. So we were trying to look to see whether the potential of the soil to break down carbon in this soil was related to the treatments that were applied, and these were on a big scale. We had 3 farms that we were doing this on. So I'm not going to go through what the effects were of the treatments, but what I will say, we did it twice. We did it in winter and summer, and we got a different answer at the 2 times. So it's not that that means it's wrong. It just means that when you do these sorts of studies, depending on when you do them, did it rain yesterday or has it been dry for weeks, the answer will be different depending on when you do it. And that's partly because this is a dynamic system, and through a season, these organisms are coming and going and doing their thing. The state of carbon is coming and going. So you can't just go out and measure something at this level of detail that will apply across a whole season. But what we like to think is that this sort of study can help in understanding what's going on, understanding processes that are involved, and that's what we are doing. Now, that isn't that useful for you. That's not, this level of detail is not a useful soil test measure for you to then make a big decision about how you're going to manage your farm. But it is useful in helping us to understand how things change over time, what implication adding different things to the soil will have. And that's why we do it. It's not necessary for you to do it. Now, if I go to the same sort of process where you do the DNA analysis for bacterial communities in 3 different soils, in this case, we added biochar, we added a lime, clay, biosolids compound that was prepared by the Water Corporation in WA. They made this product so it was safe to add to soil at one point. They're not making it at the moment. And then we had a biochar mixed with this lime, clay, biosolids material. So that was the basis of our study. Applied to 3 soils, sampled for DNA, and this time we used the DNA sequencing to say who's there, what organisms are present, and you notice that if the top 2 bacterial groups are dominant in all 3 soils. And this is common. Mostly in these soils you find there are groups which are common, a couple of groups. But in each of those little bands, there are lots and lots and lots of of different groups within it. So this is very crude. Even though it's molecular, it looks like it's detailed, this is very crude because there are a lot of different organisms in each of those bands, and they can do different things. It just happens that in this case, the dominant organisms, bacteria in this case, a lot of them are involved in nutrient cycling, and that makes sense. But there are some organisms that occur at low levels in the soil that can have a big impact on cycling. So when you're looking at these DNA patterns, it sounds really good, but it's almost like too much information and not the right sort of information for practical use, but it is useful for understanding. So this is the sort of thing we do. Now, if you pick a couple of those groups of organisms and you say, well, how does the plant affect that group? And how does adding nitrogen, in this case, affect that group? And if you look at this, the big sector here isn't changing much. This is relative abundance of the organisms in these sectors. So the dominant organism here isn't much affected by which plant it is or by whether nitrogen's added or not. So that's one story. Then you go to a different group of bacteria, and this group is affected in relative abundance by the plant present and by adding nitrogen fertiliser in this case. So the story here is that some management practices affect some groups of organisms. So the management might be the type of plant or the input, and then you have other organisms that are not affected by those. So it's a complex system and a lot going on, and it's changing with time. So we're starting to get a big window into this, though. This wasn't possible to understand prior because these things couldn't be grown. We couldn't study them in this way. But this is what's going on in soil, that this dynamic is happening. Now, I worked on mycorrhizas, and these are roots, these are mycorrhizal roots. You can't see the fungi in them unless you cook them and get rid of all the content, and then you stain them, you can see the fungi. And in this study, we were looking at the effect of adding different kind of fertiliser, adding in microbial inoculants to the colonisation of roots by these fungi. And similar to what I've just shown you for the bacteria, we looked at the relative abundance of different mycorrhizal fungi. And remember I said before that different mycorrhizal fungi can go into the same plant. In this case, ryegrass and clover, or back to front. So we were interested to see if there were more of these fungi in one plant than another. And this did happen, that the fungi all went into both plants, but the amount in the roots was different in this case. So it could matter, it may matter, but it's hard to say from this sort of investigation how these fungi are functioning. So we know they're there, what are they doing? And this just highlights some of the changes that happened, and in this case, this fungus was present in clover, but it wasn't present in the ryegrass. And we don't know whether that matters or not. So just to finish, because someone's standing up now, and that means I've got to shut up. But just a couple of things. We've done a lot of work with farmers trying to help you and others to look at your own soils and what can you test on your farm. And this little booklet we prepared with the Wheat Belt NRM, and we'll give you the link to that. It's something you can do. You don't have to send your soil away to someone else to do. And we've done a lot of work thinking about what you measure in soil that is actually practically useful to you, and perhaps that's another story, but this is a little window into something that you can do, or children that you know could get involved in these sort of activities, and that's available to you. And I already mentioned the Soil Health app, and that's a store of information that's related to this as well. It's got little videos and podcasts and information. That's it.